Good afternoon or good morning for all of our participants joining us from around the Atlantic. My name is Marie Smith from the Council for Scientific and Industrial Research of South Africa, and I will be the moderator for today's workshop. The workshop is titled Community Input, Recommendations for Monitoring the Effects of Climate Change and Emerging Pollutants in the Atlantic Aquaculture Sector. This workshop is a collaborative effort that's hosted by the CSIR, the Air Center, and consortium partners from the H2020 All-Atlantic Ocean Sustainable, Profitable, and Resilient Aquaculture, or Austral project. Okay, so just a quick overview of the workshop agenda and format for today. We will be kicking things off with a short overview of the Austral project by our project coordinator, and then we'll be moving on to showcasing some of the provisional research results from the project. Now, these are broken down into regional investigations of the various emerging threats to Atlantic aquaculture sector, focusing on climate change, harmful algal blooms, pathogens, and microplastic emissions. The results from these different research topics will also be incorporated into a publicly available report by the end of the project, which will be the Austral monitoring program and recommendations for dedicated monitoring. Just a few comments on the workshop format. During the course of the workshop, we will ask that you please type your questions to the different speakers into the webinar chat, and we will try to address as many as we can during the course of the workshop. This is, however, not just a one-way conversation, so we will be asking you all to please participate by making use of the Mentimeter or Menti application. You can access this by using your smartphone or a web browser, so you just go to the website and insert the code. This interactive session will mostly take place after the presentation, so we will give you these login details again at that time. Please note that the inputs on the Menti platform will be anonymous, but they will be captured and incorporated into the final Austral monitoring recommendations report. Okay, that is it from my side for the welcoming part of the slides. Okay, I've stopped my screen sharing, and I will next off hand it over to our project coordinator, Dr. Elisa Ravignan, who will be talking about the Austral project. So just a brief introduction. Elisa is the chief scientist at NORSE and is the scientific coordinator for the H2020 Austral project. She has a master's and a PhD in environmental sciences and more than 20 years of experience in data analysis and environmental modeling, studying the effects of human activities on the marine environment, as well as assessing all dimensions of sustainability, environmental, social, and economic with interdisciplinary approaches. Take it away, Elisa.
Thank you, Marie. I sense to be that I have a problem in sharing in presentation mode. No, it's gone, right? You see now in presentation mode.
Yes, we do.
Yes,
and we hear
perfect.
You.
Yeah, thank you so much for the introduction, and welcome to everybody that is following our webinar, our workshop today. Very nice that you could join us. So for my task in this workshop is to give you a little bit of overview about the project. And then, of course, we are going deep in the presentation about what we are doing in one of our work packages about the climate change and emerging pollutants. So short introduction about Austral that is extremely big, so it's not an easy feature. But anyway, Austral started in 2020. I will finish next year, towards the end of the year. Is a project dedicated to the whole Atlantic Ocean. So one of our tasks is, our goal is to implement the blame statement between the agreement between Europe and South Africa and Brazil, but also including Argentina and Nigeria in our case. So it's a really very big collaborative project with a lot of topics, a lot of partners. As you can see from our map, we have partners distributed along all the Atlantic Ocean. and included research at a technology organization, universities, so both academia and research, but also industry, SMEs, association and industrial clusters, and governmental and intergovernmental organizations. This is to just to set the scene about how wide our consortium is. A couple of words about IMTA that is coming more and more when we are talking about aquaculture, but maybe is not so widely known for the people not working in this topic. What is IMTA? IMTA is a way of producing aquaculture products or seafood, putting together one species or one of several species. Let's go for the simpler one. One species that is fed and other extractive species at lower trophic level that basically thrive from the uneaten feed and waste from the first species. It can be set up both in open systems, so a cage in the water with the other species along the transient along the gradient of the nutrients, or also in closed system, where it's more efficient, but of course is more costly. And one of the goal of Austral is exactly to implement a new value chain for this type of production. And how we want to do it is through our case study that we call IMTA Labs, so Austral IMTA Labs. We have 4 established IMTA Labs that are Ireland, South Africa, Scotland, and Brazil, and one potential IMTA Lab that is Argentina, where there is not an IMTA Lab yet, but they are doing a lot of research to identify the best setup for the area that is the south Tierra del Fuego, southern part of the country. As I say, there is a mix of all our IMTA Labs are different, or as a system or as a species. We have 2 open systems that are Ireland and Scotland. One flow through that is South Africa that can also doing a lot of research about at least partially recirculating, and one recirculating on shore that is Brazil. All the species that are growing those IMTA are local and anyway adapted to the area, so there is no imported species. And what we are searching is the exact to identify the best setup for those chains working together at the same time, and to develop welfare indices, so health management, to find the best biosecurity, that is one of the topics of today. And of course, it's very important, the profitability. So also the market value is assessed. And there is a lot of transfer of knowledge between our IMTA Lab to try to grow together, because sometimes, even if they are different, some of the challenges are similar. Going ahead, Austral, of course, is based on sustainable aquaculture, and it says also in the name. And so what we are doing in the project is to, assess the different components of sustainability. So the environmental one with the zero waste approach. We are also performing life cycle assessment of our chain and the circularity assessment. We are also performing the economical assessment with the cost benefit analysis and the business. We are creating business models. And also we are assessing the social dimension with evaluating the social awareness and acceptance of this type of production, and also a deep understanding and status of the legislation and regulations. I will not go in detail about, of course, we are not operating in a bubble. So the influence to and from the environment are really very important. And this is where is now here highlighted in, circling in red. That is the climate change effect, pathogens, hubs, and microplastic that will be the topic of this workshop. As I said, we are not operating in a bubble, so we also need to measure what we are doing, how is the situation in our IMTA lab, but also the exchange with the environment. So we are also developing a pool of technology innovations where through biosensor and spectrometers, IoT kits and vision sensors, we plus, of course, other tools already available, we are able to understand better the water quality, the environmental, the animal welfare, the psychochemical variables, and some estimations like biomass estimation and the hub monitoring. But of course, what is important, and Astral is definitely dedicated to that, is the collaboration between the two sides of the Atlantic Ocean, but also from the north and south. So the collaboration, for example, the first collaboration that comes in mind is the collaboration with the stakeholders. We have a collaborative community that we call Atlantic Aquaculture Network, where we can discuss challenges, solution, and business opportunity. This is done through our Aquaculture Helix platform. That please get in contact with us if you are interested in this, to be part of this platform. We are also building capacity, and education is also very important for the project and for the area. Also, if we want to really establish a sustainable aquaculture. So we are developing a human capital development plan that is focused on regional and scientific and societal challenges, especially in the area with the low, medium income. And we are doing that through apprenticeship, summer schools, and short technical courses. We have also, through our partner in Nigeria, a huge, very focused task on women empowerment in aquaculture. We have done a lot of work there. Please follow our social media. You will find a lot of interesting work done on that topic. And of course, we have extensive collaboration with the other projects, not only in our own call, so our twin project Aquavita, but also all the other along the Atlantic Ocean and a lot of others working on sustainable aquaculture. I think I can stop here. Here you have all our social medias, and of course you can find it also online. Please follow us, engage with us, not only today, but also afterwards. We are always happy to receive emails, questions, or comments. And please, Marie, go ahead with now the real show that is about the topics we want to discuss today. Thank you.
Thank you very much, Elisa, for giving us such a comprehensive overview of the Astral project. Next up, I'm handing over to Dr. Nadine Gores, who's talking about climate vulnerability along the Atlantic coast. Just a brief bio, Nadine is a researcher in climate science at the Bjerknes Center for Climate Research in Norway, working on the research topic, past, present, and future climate trends in ocean biogeochemistry. She is also currently the PI of a strategic project of the Bjerknes Center, where they try to create more certain projections of future trends in dissolved oxygen. Take it away, Nadine.
Yes, thank you. Very nice introduction. Thank you very much. Yeah, so I'm going to talk about the climate vulnerability along the Atlantic coast. This is work that had been mainly done by Augustine Kessler, but in cooperation with me and Siv, who's also virtually present. So in case that there are questions, she can also jump in and help me with answering them in case that I can't. Yeah, so for this task, we try to establish climate trends, and we put the focus on large marine ecosystems along the Atlantic coast. And these you can see on the right side in the picture in the light blue, and with black there are borders. And I also put the IMTA labs there. You can see this in red. These are the IMTA labs that we are considering in Astral. or it's a place of a potential future Imtalat. And we are establishing these trends by using in situ observations from large databases. These are SOCAT, the World Ocean Database, and GLODAP. And the fact that we use these in situ observations is also where we kind of differ from most of the studies that we've seen, because other studies have mainly been using reanalysis products or model products. But we think that now there are enough observational data available to estimate these trends with in situ observations. And we do this for the following parameters: sea surface temperature, sea surface salinity, oxygen, nitrate, pH, and partial pressure of CO2. Before I'm going to show the results, I'm going to speak a bit about the methods that we are using. So first, I'm going to speak about a common problem that occurs with relatively sparse observation, that is the spatial aliasing. That means that simple averages of observations can be biased towards areas with the highest density of observations. And here you see on the left side a schematic of a large marine ecosystem, where the blue is supposed to be a lower temperature and the red a higher temperature. And the black dots are observations, and you can see if I have fewer observations where it's colder and more observations where it's warmer, and I simply calculate an average over it, then my average will tell me that the ecosystem is on average larger than it actually is. So we correct this by adjusting the observations with their spatial gradient, so that the adjustment centers each of the observations toward the center of the large marine ecosystem, so that it doesn't matter so much if there's a spatial aliasing present or not. The other adjustment that we do is related to the temporal aliasing of observations. That means that observations are usually also not distributed evenly across months, but they might be more frequently measured in certain months and less frequently measured in other months. For example, when there are winter storms or other things happening in an area, then it's less likely that during these months these areas are frequently observed. So this temporal aliasing we correct by mimicking a seasonal cycle with a cosine. And here you see, as an example, the SST data in blue for the large marine ecosystem that is Patagonia. And you can see that if we make an annual average, but only there where all 12 months are presented, then you can see with these green stars that especially in the early years, there are not all months presented, so we are not able to form an annual average. And in order to still be able to calculate trends that are not biased towards certain months, we use this cosine approximation, which you can see in gray, to kind of estimate the annual amplitude. And then with this cosine approximation, we calculate the trend. Another thing that has to be considered is that the data coverage is, of course, not everywhere exactly the same. So dependent on the large marine ecosystem, but also on the variable that we are considering, the data coverage might be less or more. And here you can see for sea surface temperature, an example for several of these large marine ecosystems, where you can see in the last row how many of the months between the years 1957 and 2020 are actually covered with observations. And you can see that there are some large marine ecosystems, for example, the Caribbean Sea, where really all of these years and all of these months are covered by at least one observation. But there are other large marine ecosystems, like here, for example, the East Brazil Shelf, where we only cover 23% of these years and months. And that also means that the trend estimate will be less confident. So here you can see the results for sea surface temperature for the larger time span. This is from 1957 to 2020. And you can see that the results are can be quite diverse, with some ecosystems showing really a large increase in the average temperature and some showing only a moderate increase. Additionally, we also looked at only the last 40 years of this period. This is 1980 to 2020, and compared it with a longer time period. So here you see the longer time period in dark gray, the shorter and more recent period in light gray. And the more the difference is between this dark gray and the light gray, the more the trend is accelerating with climate change. And here you can see that there are some areas, for example, the North Sea. This is the second large marine ecosystem from above, where we see a large acceleration, whereas there are others where we don't see such an acceleration at all. So based on these results, we estimate that the Scotland and Ireland MTERs, which are in the Celtic Biscay large marine ecosystem, they have a large acceleration of SST in the recent years and therefore are more vulnerable towards this, whereas we don't see the same signal in the Brazilian MTER, on the Beagle Channel, or in the South African MTER. We also did the same kind of analysis for other variables, that is sea surface salinity, oxygen, nitrate, pH, and partial pressure of CO2. Here you have to note that the data coverage for oxygen and partial pressure of CO2, or also pH, make a confident trend estimate difficult. And so the results that we had here, they had to be confirmed with literature, and that of course makes the results also less novel, and then again also dependent on reanalysis or other model studies. And the data coverage for nitrate did not allow for a confident trend estimate at all. So what we have now for the MTERs that we are considering in Austral is that we think that Scotland and Ireland are very vulnerable towards increasing SSTs. But for sea surface salinity, we see no alarming trends. Also not for oxygen. Though oxygen is declining, and it's declining faster where the SST is increasing faster, the trends are not yet concerning for each of these MTERs labs. For nitrate, we cannot estimate a trend because of the data coverage. And for pH, we see a faster decrease in, again, Scotland and Ireland, also in the Brazilian MTER, less of a decrease in the Beagle Channel and the South African MTER, but still a decrease. So here, all MTER labs are vulnerable for a pH decrease. And of course, coinciding with this, we have a PCO2 increase, which is maybe as a variable in itself not so interesting, but only in relation to pH. And that is already my presentation. Thank you for listening. Yes.
Thank you very much, Nadine, for sharing all those interesting results with us. Next up, we're going to be looking at conditions favoring harmful algal bloom development, which is going to be presented by Dr. Franziska Vermeulen. Franziska is a postdoc in algal biotechnology at the Scottish Association for Marine Science. She has an interest in cultivating microalgae for a variety of biotech applications and is currently involved in assessing environmental factors associated with harmful algal blooms and identifying future risks and impacts. Her previous research focused on optimizing cultivation conditions for commercially important microalgal species, and she has experience working in an industrial environment with commercial scale algal cultivation systems. I think Franziska is Natalia, are you going to share the presentation?
Yes.
Okay, cool.
Yeah. Thank you.
Just waiting a few moments for the presentation to be shared, and then Franziska, you can take it away. Please remember, for everybody, if you've got any questions, you can type it in the Q&A box to either Elisa or Nadine so far. And then obviously, as all the rest of the presenters give their talks, you can ask them questions as well. Okay.
Yep. So for the Astral project, I've been looking into conditions which are favoring harmful algal blooms and how we can monitor and get a better understanding where and when they occur. So next slide, please. So this presentation, I'll just quickly go over which factors have been identified that contribute to harmful algal blooms, some monitoring recommendations and some methods we can use for monitoring, and possibly also some schedule or on which time scales we want to do the monitoring. Next page, please. So harmful algal blooms is not just one algal species. There are many different species, and some will occur almost across the world, while others are very region specific. And that makes monitoring and understanding harmful algal blooms more difficult. Some species might be toxic at very low cell concentrations, others form massively dense blooms, or have spines which can harm fish. So there are different life strategies, different sizes, shapes. So one of the most commonly used way to monitor for harmful algal blooms is just to measure chlorophyll, because all harmful algal blooms will produce chlorophyll, but of course all algal cells will produce it. So you know there are algal cells present, you just don't know if they're the harmful ones or not. So secondary, we also use microscopy. However, this is very time consuming, it's very slow, and it's a very highly skilled job to do. So we want to have more methods for measuring and understanding harmful algal blooms. So next slide, please. So you also want to look in environmental conditions. And one of the big factors we notice from harmful algal blooms is that nutrients play a large role, and especially the ratio between the different nutrients. So in generally, the harmful algal blooms are dominated by dinoflagellate species. And when nitrate increases, those species will also increase. While, for instance, when silica is increasing, you'll get more diatoms. And though some diatoms can be harmful, in general, they're less harmful or associated as more beneficial microalgae. So it's very important to measure all those different nutrients as well as dissolved matter, because a lot of the dinoflagellates can actually take up dissolved organic matter as an alternative food source. So that will cause, for instance, blooms of Carenia and Alexandrium, which are very toxic. So we highly recommend measuring nutrients and the ratios between them. So next slide, please. So other than nutrients, you also want to think about oceanographic conditions. And we already saw a lovely presentation earlier where they were monitoring sea salinity and temperature and stuff. And these play a really critical role in monitoring for HABs as well, especially things like temperature and stratification. Quite often blooms are associated with stratification. So that means if your waters are warmer, they're stratifying, your likelihood for HABs to occur will increase. While when it's mixed well, the water column, then there is a less likelihood. It's not meaning that it can't happen, but it's less likely. So understanding how those different oceanographic and meteorological conditions contribute to the likelihood is very important. So understanding, for instance, the wind can affect the timing and can also move blooms to other locations. Upwellings can introduce new nutrients, similar as runoff, and high precipitation can cause increase in nutrients. So understanding these variables are very important. Furthermore, I think it's very important to understand your own region. So there are some major contributors like nutrient, temperature, and stratification. But in your own region, some other factors might play a big role as well. For instance, in Argentina, we see glacial melt, which can have an effect on your stratification and harmful algal blooms. So monitoring that can be beneficial. Other areas, for instance, in the Gulf of Oman, you'll see that eddy movements are really important in understanding where blooms are and where they're going. And in Cork in Ireland, the tidal cycles have a very big influence on when blooms occur. So besides the general recommendations, really think about your area and what could be beneficial. Next slide, please. So of course, we can take samples, we can look under the microscope, we can do measurements, but they are just one-point measurement. So Earth observation data using satellites, as earlier presented, can provide a very nice, very wide-ranging measurements of salinity, chlorophyll, temperature. They can be done over a wide area, they can be done continuously. So that will provide a much better data set than just a single sample. Samples still play a big role just to get extra information, but combining these measurements and using Earth observation data, we can actually start making models in seeing how blooms behave and perhaps also make predictions of when blooms will form or where blooms might be going towards if they're formed offshore. Next slide, please. So we have a list of recommended monitoring. So the chlorophyll, of course, is an indication for your microalgae to be present. This can be done in the field. This can be obtained through satellite data. Of course, we need to identify them. And then the main ones to monitor as well is nutrients, salinity, currents, upwelling, stratification, and temperature. These can be collected in multiple ways. Of course, it will be too much to do all those measurements all the time in all circumstances. So we also develop roughly a time scale or a suggestion on when to monitor these. Next slide. Yeah. So for the time scales, chlorophyll, if you use satellite data, that can be more often. Otherwise, maybe monthly would be better. There's also equipment which can be placed in situ, which can monitor on a continuous basis. So there's a large variety there. Hub species, most monitoring programs are just once a week, which seems to be sufficient to get an idea of what hub species are present and give warning to aquaculture sites or IMTA sites to tell them that there's harmful species in the area. Nutrients tends to be done monthly, salinity similar. Current can be done by satellite on a more regular base, and temperature is also one of those variables which can be monitored continuously or just on a weekly basis. And once you have identified which parameters are important in your area, try to increase those monitoring timescales for those particular ones. Okay, next one, please. And that's the end of my presentation, and I hope you get a better understanding of all the different factors contributing to hubs and which might be of interest to monitor in your area. Okay.
Thank you very much, Francisca. Right, so all those conditions favoring harmful algal blooms, if you have any specific questions, you can please pop those in the chat for Francisca to answer or in the Q&A session. Right, next up we have pathogens, past, present, and future, which will be presented by Marina Martin Sandoval. Marina is a scientific researcher at the LETAT Technological Center in Spain. She has an MSc in biotechnology from the University of Aberdeen in Scotland and great interest in the study of microorganisms and their interactions with the environment. She is currently working at the Applied Microbiology and Biotechnology group at LETAT, where they aim to better understand these topics and make a positive impact in the field. Right, I'll hand over to Marina. You are not muted. Cool, thanks.
Hi, thank you. Can you hear me?
We can hear you, just can't see your presentation.
Yeah, just sharing it now. Can you see?
Yeah. almost in presentation mode. There you go. Thank you.
Okay, thank you so much, Marie. Okay, so nice to meet you all. Thank you so much for joining the workshop. My name is Marina, and as Marie said, I'm working in LITAT as part of the Applied Microbiology and Biotechnology group. And today I will be talking about pathogens in the IMTA system and how, based on literature review and based on the emerging technologies, we aim to understand these pathogens better and how they work. So aquaculture, as we know, is a fast-growing food sector, but this also implies that there are associated challenges and risks to the production and the growth of aquaculture. And this means that there's diseases, and all species in aquaculture system are susceptible to these diseases. They can be caused by different pathogens, such as bacteria, viruses, and other pathogens. So these outbreaks are expected to increase due to climate change, open water being the most vulnerable ones. So we aim to understand and monitor these outbreaks better so we can prevent them rather than tackle them once they have occurred. So our aim is to describe these pathogens and their identification of the emerging technologies to improve the monitoring methods in the IMTA labs. So basically to be able to prevent pathogen outbreaks before they happen. So basically what we did, or what we've been working on, is describing the most problematic pathogens and the early warning signs that we have and that they have been showing. Then we've been working on real microbiome data from Astral IMTA labs. And then with this, we've been researching emerging technologies from literature to be able to understand how outbreaks happen and to be able to catch them in time before the disease is already there. So firstly, we looked at the most problematic pathogens described in the literature for the aquaculture systems, but also we looked into the environmental characteristics, and we made a plan to monitor this in aquaculture. So the current methods for controlling outbreaks include antibiotics and vaccines, and also control of the environmental conditions, but this is mainly focused after the disease has happened. So we focus mainly on bacteria, virus, but also on other pathogens like sea lice. And we realized that there's a high need to improve and develop technologies that prevent pathogenic infections. So firstly, we need to talk about how disease happens. And this happens because it's important to understand that disease doesn't only occur because of a pathogen, but also because of the environment surrounding it in the aquaculture system and the susceptibility of the host. So if the host is under stress conditions, or if the environment is likely for a pathogen to occur, then it's more likely to, there's more likely to occur an outbreak. So we don't only want to see the presence and absence of this pathogen, but we also want to take all of these conditions into account when monitoring aquaculture systems. For this, currently there's both molecular tools and biosensors for both pathogen detection and environmental monitoring and surveillance. So mainly there's, for pathogen detection, there's mainly molecular tools such as PCR, high throughput sequencing for the microbiome analysis, qPCR. But the downside is that this equipment is very expensive. It needs processing of the sample and it cannot be used easily in the field. On the other hand, we have biosensors that allow also for pathogen detection and environmental monitoring. But the downside is that it needs to be more specific to target different pathogens. In literature, we've seen that the pathogens that we have chosen have been used or have been like targeted by different molecular methods and biosensors, but we see that there's a limitation to both of these methods, as I mentioned, and that there needs to be a combination of these two methods so that we can use biosensors in the field to easily detect and prevent outbreaks. And then that we can use these molecular methods to develop on this information that we get. So future technologies for the detection and monitoring of pathogens in IMTLab should focus on a combination between molecular tools, biosensors, and also microbial data. So we can develop prediction models. Although molecular tools provide specificity and accuracy, they require processing and careful handling of the samples. So it's easy to take a lot of time while you're using them. And although biosensors are cheap and fast, they need standardization for researching pathogens. So we aim to improve the early diagnosis with monitoring of the aquaculture systems in real time, so that by monitoring the environment, we can predict outbreaks before they actually happen, and we can see the early warning signs without having to use antibiotics or vaccines that are after our disease has already occurred. We aim to improve this system and avoid post-infection treatments that are becoming obsolete. So thank you so much for listening, and yeah, I'll stop sharing my screen.
Thank you very much, Marina.
Thank
That was
you.
very interesting. If you have any questions for Marina, please pop them in the Q&A session. Right, next up we are going to listen to Dr. Alessio Gomiro from NORS on microplastics in aquaculture systems, past, present, and future. Some background: Alessio is a senior scientist at NORS, specializing in chemical quantification and characterization of polymers in environmental matrices and their associated biological effects on model organisms. Alessio leads research initiatives that are developing methods to estimate micro and nanoplastic occurrence and composition in marine, freshwater, and terrestrial environments. He is also the scientific coordinator of the North Atlantic Microplastic Sensor and a member of several expert groups dealing with plastic pollution. Take it away, Alessio. You are unmuted, and we just need to see your presentation.
Okay. Thank you, Laurie. Thank you very much for this nice introduction about myself, and thank you everyone for attending this presentation. So I'm going to talk about microplastic, the microplastic pollution associated to the aquaculture system. What we know about the plastic? Well, about the plastic, we know that we have a plastic everywhere, has been like detected in every corner of the world, including also the abyssal trenches and also the Icelandic glacier, for example. Several studies have shown that when you release a large plastic item into the water, it goes into fragmentation, and then micron-sized plastic fragments obtained from a large item may also enter into the food web. So it can be like accumulated in different marine species, including also those of great ecological and economical value. There's been a lot of different attempts try to characterize and map the different emissions associated to large industrial activities, including also aquaculture. But at the moment, it's really limited, the knowledge we have about the different type of emission and the plastic fingerprint associated to the aquaculture. Related to aquaculture, of course, plastic is a fantastic material. You can shape in different way, you can shape with different color, and you can give also different type of properties. It has replaced the natural type of material in time because it is light, it is technically strong, and it is doable. But when it goes into doable, then we start to have a lot of problem because it persists in the environment. So we postulate that using so much amount of plastic in aquaculture, most likely there will be a significant release. Related to aquaculture, lesson learned from other type of project, we have been also identified that possible sources of plastic may or not only come from the production per se, may also come from the food, may also come from the different processes that, for example, deliver the food to the different nets. So it is a 360-degree angle type of problem, the occurrence and the release of plastic material. In a straw, we have been like developing different type of prototype to accurately sample different type of sample. You may see an example on the back part of this presentation on the left side. And in Astral, we focused mainly on the mobile phase, so the water column, and also on the sediments, collecting both water and sediments across the different case studies. RAS system actually is even less known what is actually happening into this type of production. We know that water is collected from a reservoir, from the sea, and then it is circulated several times before being released if we are talking about a semi-open recirculating system. But so far it's very difficult, and there's not a lot of literature associated to the occurrence, the distribution, and the concentration of plastic particles in a different step of the production process. In Astral, we have been like focusing on different case studies, both recirculating system and semi-recirculating system, and also open system located in Brazil, South Africa, Scotland, Ireland, and Argentina. As you may see in this picture, we have been already running different sampling activities using the ad hoc designed sampling device, and then different type of sample preparation has been performed in order to release all the interference from our matrix and then be able to characterize the plastic content in our sample. As you see here is like a multi-step enzymatic and strong oxidative reaction, which is selectively taking out all the different fractions of the organic matter, which may target or interfere with analytical equipment that we are using in Astral for the characterization of the plastic particles. The type of technique we have been using for the characterization is a vibrational microscopy oriented technique called the micro FTIR, micro Fourier transformed infrared spectroscopy. As you see visually here, what we have been like collecting and obtaining from the sample preparation is deposited on an infrared compatible window. It is then scanned through a micro infrared to collect images about the filter. And then this image from the visible image is converted into infrared one through the eye of the infrared camera. And then the resulting infrared peak are somehow compared with the reference of a database. And then the post-processing is basically an automatic detection system called SIMPLE, which basically reflect and report the shape, the size, and the polymeric composition of every plastic particle that has been collected within our samples. At glance, I'm not presenting all the results we have been like obtaining so far, but I'm reporting somehow the averages across the different type of EMTA system, both open and closed. Typically, what we normally assess in Astral is the total number of plastic particles, the relative abundance of the different polymer, and the different particles associated with the polymer. Then, to move from the actual observation to the future trend, you may understand that we need a well fine-tuned modeling approach. In Norse and in Astral, we have been like developing in time competence in modeling such complicated type of particle into the aquatic environment, obtaining a fine, high resolution, local level type of model for the distribution, which also is attempting to match to the distribution at a global scale. So in the future, after the final round of assessment associated to the occurrence of plastic particles in the different EMTA, mathematical modeler, we will try to simulate their distribution, the final fate into the environment, and according to the general trend, also draw possible conclusion about a possible hotspots associated to the activity related to the different EMTAs. Thank you very much for your attention.
Thank you very much, Lisa. It's always very interesting to hear all the work happening in the microplastics realm. While we wait to see if there's any more questions for, or if there's any questions for Alessio in the chat, Alessio, can I maybe ask one for you?
Sure.
It's out of my personal realm as a remote sensing scientist. Do you think there's any scope for there to be use for satellites in monitoring microplastics potentially in the future? Or do you believe the in situ data is always going to be by far the best?
This your approach is actually already somehow a reality in pilot, because the interaction with the satellite observation, it is already ongoing in different type of research. Actually, some of them I'm also in contact with. So in the future, it will be possible to monitor the floating litter through satellite without any problem, let's say with a reasonable resolution. So we may be able to somehow characterize the distribution of the mesoplastic. So fragments up to 1 millimeter.
Okay. Cool. Thank you very much, Alicia. Right, so we have a couple of questions in the Q&A session. We had one that was passed on in private to Francisca. Francisca, I don't know if you have had a chance to see each one, but I'll quickly ask it to you out loud if that's okay. The monitoring recommendations that you said are applicable to all geographic areas. Are they applicable to all geographic areas, or are there parameters that you would recommend monitoring on a different scale? I'm not sure if that's spatial scale or temporal scale.
The ones at the end of the list, I would recommend that is just everywhere. These are the most commonly most important ones. So things like salinity, chlorophyll, nutrients, those are just, let's say, universal. And in addition to that, you have to look into your own area, whether you have a lot of runoff from rivers, upwelling, stuff like that. But the final ones, I would say that is pretty much universal, yeah.
Cool. Thank you very much. I can maybe just see the pathogens questions were quite popular. Or the pathogen, there's a lot of interest in the pathogens. I can maybe just ask the last one, which was, what are the emerging pathogens that users in the field should be aware of? Any specific species or groups for fish and or shellfish? Marina, I know you've typed it, but maybe we can just go through it in person quickly.
Yes. Yeah, so my answer was for example, Atlantic salmon and lumpfish, bacteria like Armona salmonicida, and also beaver species is really prevalent in a lot of species, so that's really important. As for virus, for example, for shellfish, there's abalone herpes virus affecting mainly abalone. And it's really important to monitor them because they usually die within 1 day after infection. So it would be really helpful to tackle it beforehand. Yeah.
Oh, thank you. Okay, so I think while we still have quite a few people online, I'm gonna ask that we head over to the Mentimeter application. I'm just gonna give you guys a quick overview of how that works again. Okay, hopefully everybody can see my screen. So Mentimeter is a platform where you can get interactive feedback. For you to access the questions, we can either scan the QR code with your phone's camera, or you can visit the website either using a browser on your PC or your mobile device, and just enter the code 54976241. We'd like you to please answer all the questions if you are able, and only skip a question or a sub question if you really don't know the answer to it. And then also remember to press submit after each of the questions. The questions are only going to change once I actually click forward. So I'm just going to stay on the same question for a little while to give everybody a chance to respond. Okay, so I'm going to stop sharing this application. There will be some information on the chat on the code that you need to enter in case you missed it. But I'm going to stop sharing so we can actually see the results of the application. I see we've had quite a few people already on there. So I'll just quickly share that. Okay. Okay, so the first question that we are asking is what best describes you? And this is in your personal capacity, whether you are an aquaculture person, whether you're a researcher, a regulator, a policymaker, or a student, or if you identify as other, if we didn't capture you in any of the other ones. Okay, Natalia and everybody, I can't monitor the chat right now. So if there is any questions of how to get to Menti, can you guys please just assist there? Okay, so clearly we've got a whole lot of researchers that are attending our presentation today. That might just be due to where we've been sharing this, workshop. Okay, I'm going to give you guys just a little bit more time to actually get onto Menti. I know once you're on there, it's easy to answer, but it might take a few minutes for people to get yourself there. Okay, I can just write the code again. It's 54976241. Sometimes it is easier just to use the QR code on your phone. Then you can access it directly. You don't have to input any of the codes.